Hello friends, Ace Votain, next episode. In this episode, we'll be discussing YTQ question series for NEET exam. Today, it is 1-5A. Why I put A? Because today's chapter is Principles of Inheritance and Variation, first part. Second part will be in the next episode, that is 1-5B. Because we cannot discuss too much questions in a typical or a class. Duration will be there. That is why I put up to chromosomal theory of inheritance. Almost 30 questions connected with NCERT. Practically seems to be a good question. So we can proceed. I will share the screen so it will be convenient for you. I think screen came, so we can proceed. So first one, humans know from as early as dash that one of the cause of variation was hidden in sexual reproduction. So up to how many years back we know it? It is almost 8,000 to 1,000 BC. So answer-wise, it is 3. And it is direct from NCERT. So left side, I put some photograph, Mendel, Poisson sativum, 7 characters, all this. That is for immediate reference to you. So no need to go back to your textbook or material or what may be. So answer-wise, it is 3. And normally, sometimes after each question, there will be certain writings on the bottom of the page. So that is an important item connected with the question that you have to read. That is why I separately given it. So first question, option is 3. I think idea clear. NCERT page number also given. Now we can proceed to second question. Through artificial selection and domestication from ancestral wild cows, variation can be exploited which were naturally present in them, as in the case of, again, NCERT-oriented question. It is Sahiwal cow of Punjab. NCERT page number is given, 54. Photograph is also shown there. So answer-wise for question 2, it is 2. Direct question. Now question number 3. Mendel finally selected how many characters? True breeding. By some varieties located in this characters located in how many chromosomes? Now in each chromosome, how many characters? That way the question goes on. It's a good question. So we have to take care many options we have to choose. So Mendel finally selected 7 characters, even though he started from 31, 32 characters initially. Now finally came into 7 characters. And each with 2 different traits. So that means 14 true breeding varieties. So 7 and 14. And these characters are located on how many chromosomes? It is located on 4 chromosomes. And chromosome numbers will be 1, 4, 5, 7. And respectively, characters will be 2, 3, 1, 1. And this kind of study was done by a famous scientist, S. Blixt. And what are these characters in which chromosome it is located? That is what I shown on the bottom of it. And we can find characters are given. Chromosome number 4, 3 characters are there. That is pod shape, flower position, stem height. And genes for pod shape and plant height is closely packed. That means there may be linkage between them. So please read bottom that red mark or what the color that the statement given. It's important. Connect with that question. So I think for question number 3, answer is 1. It is clear. Hoping so. We can proceed to next question. Question number 4. In a cross between pure tall plant and pure dwarf plant, F1 produce. That will be tall. That is why tall is dominant. F1 is then self. F2 produce. So rule is parent. Parent is crossed. F1 produce. F1 is self to produce F2. So in genetics, that is a rule. Sometimes in question, they may not mention it. Percentage of heterozygous tall among tall is. See, we can just write here. Capital T crossed with a small t, capital T small t gamete, capital T small t will be F1. Gamete will be normally circled. F1 will be self. So when F1 is self, we can draw checkerboard. That is the easiest item. Capital T small t, capital T small t, familiar to you. This way it proceeds. So we can find phenotypic ratio, it is 3:1. Genotypic ratio, it is 1:2, 2:1. Okay. So normally in the question, the percentage of heterozygous tall among tall. So people may misguide. They need a question is required tall, and they may select 75%. This is wrong. Question is among tall. Among tall means 3 by 4 is tall. How many will be heterozygous tall? So tall will be 3. Out of tall, 2 will be heterozygous tall into 100. So that will be around 66%. I think answer is clear. So option wise, nearest one is 3. I think idea clear. And below it, normally in Mendelian cross, P is crossed, F1 is self. I think idea clear. We can proceed to question number 5. Punnett square was developed by? That is a previous question, direct NCERT. It is by Reginald C. Punnett, British genetist. So option wise, it is 4. And below it, you can find a statement. It is the graphical representation to calculate the probability of all possible genotypes of offsprings in a genetic cross. Now, question number 6. So when we come to question number 6, to determine the genotype of a tall plant at F2 Mendel cross, the tall plant from F2 with a dwarf plant. This is called as test cross. That is NCERT, page number 58. So option is 4. That is correct definition given by Mendel. Now coming into 7th one. In a typical monohybrid and dihybrid, test cross ratios will be, monohybrid will be 1:1. Dihybrid will be 1:1:1:1. Both crosses given there. So option wise, it is 2. 7th one, option 2. Now we can proceed to 8th one. So when we come to 8th one, find the odd one for Mendelian monohybrid cross. So usual question, out of 7 characters or out of 3-4 rules, postulate which is odd one. Regarding monohybrid cross, so we can find law of independent assortment came after dihybrid cross of Mendelism or Mendel. Other 3 based on monohybrid cross. So here it is odd one. Odd one will be law of independent assortment connected with the dihybrid cross. So option wise, it is 1, NCERT page number 64. I think idea clear. Now we can proceed to 9th one. When we come to 9th one, how many original parental genotypic condition, double heterozygous condition, is there in F2 Mendelian dihybrid cross out of 60? First is original parental genotypic. So that is, we can take in this manner. Full dominant, full recessive, dihybrid. So this way, we can find 2 will be there in Mendelian crosses. 2. Since it is a very small diagram, that will be at 2 extremes, we can find this one. Then, double heterozygous condition, that will be this way. This way, we can find 4 in number. 2, 4. So option wise, it is 3. I think idea clear, direct from the checkerboard. Now below it, I written some statement. Please read it. It is important for exam purpose. Here you can find for R, capital R, small y, RR side is homozygous. Similarly, small y side is homozygous for respective features. But this way, in original parents, you cannot find. Original parents will be this way only. So option wise, it is 3, 4, 9, 2. I think idea clear, we can proceed. With a dihybrid cross, how many such questions can arise based on checkerboard or Punnett square? When we come to 10th one, what is the ratio between yellow seed and green seed, round and wrinkled seed shape in Mendelian hybrid cross? So in this cross, we can find there will be 12 boxes, at least with the capital Y. Means there will be 12 yellow seeds. So remaining 4 will be with this way, that is green seed. So 12 is to 4, that is 3 is to 1. Same manner, round and wrinkled. 12 round will be the, then 4 wrinkled will be the, that means 12 is to 4 again, 3 is to 1. So 3 is to 1, comma 3 is to 1. So option wise, it is 2. I think idea clear. So we can proceed to question number 11. Type of gametes, number of phenotypes, number of genotypes in a trihybrid experiment. So what we can do here is monohybrid. First is gamete. Phenotype, genotype. So when we take number of gametes in monohybrid 2, phenotype tall and dwarf. Genotype capital T, capital T, capital T, small t, small t, small t. So it will be 3. Now dihybrid. This way progression you take. Gametes will be 4. Phenotype will be 4. And what about genotype? 9. 1:2:1:2:4:2 4:2:1:2:1. Now same manner when you take trihybrid. 8, 8, 27. So it is 3 square. Here it is 3 cube. 2^2, 2^3. This way you can take it. So it will be easy based on progression. Any cross monohybrid, dihybrid, trihybrid, tetrahybrid, any way you can take it. So option wise it is 1. I think idea clear to all of you. So based on NCERT figure, dihybrid cross. Now coming into 12th question. In a typical dihybrid ratio between, okay, ratio given. So we can find all are pure, so only one will be there. Here also all are pure, so only one will be there. Here we can find 4 different gametes can be produced, so 4 boxes will be there. Then here 2 types of gametes can be produced, so it will be 2 boxes. And this way 2 boxes, so this way it will be coming. So 1:4:2:2. That means it is 4. I think idea clear to all of you. Direct to question, simple. Now coming into 13. Incomplete dominance can be typically seen in the flower color of. It is seen in the dog flowers, snapdragon, anti-rhino, all are same. Answer is 4. Diagram also given there. So it will be easy for you. Now coming into next one. Genotypic and phenotypic ratio is same. That is, it should be like 1:2:1. Same as in 1 is incomplete dominance, right? Cross between AB individuals, AA, AB, AB, B, B. Monohybrid cross in garden B with the size of starch grain. So how many statements are correct? All are correct. All will be showing 1:2:1 ratio. So option is 1. In monohybrid cross, capital T small t into small t small t. That is called test cross. Ratio will be 1:1, phenotypic and genotypic ratio. In dihybrid test cross, it will be 50% tall, 50% dwarf, 50% red, and 50% white. So here answer is, all are correct. Option Y is 1. Now coming into 15. A single gene product may produce more than one effect. It is called. It's a famous question. See, there is a root term. Term is pleo. This means not constant. Or we can find, or we can say changing. So one word you studied in not morphology, bioclassification, that is pleomorphism, constantly changing the shape. Certain groups of organisms can change shape constantly. Pleomorphism. Here it is pleiotropism. That is single gene producing more than one effect. That is the simplest definition. So answer-wise, it is 3. And what is the meaning of the term pleiotropism? See, main, or what is the reason? Main basis of pleiotropism is the relationship between the metabolic pathways that may contribute towards different phenotypes. So option-wise, it is pleiotropism. I think idea clear. Now coming into 16. Example for pleiotropism, single gene producing more than one effect. Typical examples that all will be, maybe already studied still as a question. Drosophila white-eye mutation also lead to depigmentation of many other parts. So single gene, more than one effect. Phenylketonuria, there will be mental retardation, reduction in hair and skin pigmentation. Then gene connected with the starch synthesis, shape of seed, and size of starch grain. Single gene. So all of this. So option wise it is 4. And regarding starch synthesis as well as size of the starch grain, I put it there. So regarding starch synthesis, capital B, large starch grain, capital B, small b, intermediate starch grain, small b, small starch grain. So capital B, small b shows incomplete dominance regarding size of the starch grain. While seed shape is considered capital B, small b will be dominant. Small b will be only recessive, wrinkle. And basis of pleiotropism, I only told you in the last question, same thing I written. So please read below the question what is written. That is important for you also, informative. So 16.4, now coming into 17. Theoretically, the modified allele could be responsible for the production of, again, NCERT direct line repeated question, normal or less efficient enzyme, non-functional enzyme, no enzyme at all. Option is 4. Then 18. When F1 is resemble either of the two parents, then P or was in between Q. But in the case of dash, that F1 generation resemble both parents, P, Q, R respectively stands for. So when F1 is resembling either of the two parents, then it is called dominance. Or was in between, it is called incomplete dominance. But in the case of codominance, F1 generation resembles both parents. This is a famous line from NCERT page number 61. Same statement I put it there, but when I put it like this way, it will be slightly confusing. So option wise, it is 1. This time questions will be twisted, I think, because so your time will be lost. So when time goes means you will be perplexed. That is A. So take care. Questions will be simple normally, but this way they will put it, long questions. So 18, option is 1. I think idea clear. It's all NCERT, nothing outside. Now 19, ABO blood group gene is located in chromosome. It is not in NCERT, but we should know it. ABO blood group gene is concerned with the chromosome number 1. Chromosome number 1, which arm? It is called long arm of chromosome number 9, band 9q34. Short arm of chromosome is called P arm. Long arm is called Q arm. P means it came from a French word petit. Petit means short or small. In some mutation, we'll be learning this P arm, petit arm, or a long arm, short arm like this way. So 19, option is 1. Answer is 9. Now coming into 20. ABO blood are controlled by the gene. The gene is called I gene. I gene is in first option, and I designates for, or designation stands for isoagglutinogen, which is another term for antigen. That is why the term I came. So 20th question, option is 1. That is I gene. 21. So when we come to 21, ABO blood group system is an example for. Choose from the statement. Under normal condition, it can be example for codominance. It is an example for multiple allelism. But it is nothing to do with incomplete dominance. So 1 and 2 are correct. 1 and 2 means option is 2. So ABO blood group system is example for dominance, codominance, and multiple allelism. I think idea clear. Now we can proceed to question number 22. So when we come to question number 22, ABO blood group system total, how many genes, how many alleles, how many phenotypes in an individual, how many alleles maximum seen. This all under normal condition. So we can find in ABO blood group system, total how many genes? Only one gene, I gene. I gene. How many alleles? 3. A, B, and O, respectively. How many phenotypes? 4 phenotypes, A, B, O. In an individual, how many alleles maximum possible? In an individual, maximum possibility is 2. Then, under normal conditions. So answer-wise, for 22, it is 2. I think idea clear. So 1 a gene, 3 alleles, 4 phenotypes. In an individual, maximum 2 alleles. So option-wise, it is 2. Now coming into 23. In ABO blood group system total, how many genotypes will be there? So we can find there will be 3 alleles. When multiple alleles are given, to find the genotype, there is a simple formula of n into n plus 1. So value of n is here 3. So half into 3, 3 plus 1. Half into 3, 1.5. 1.5 into 4, that is 4 plus 2, it is 6. So option wise, it is 3. 23, I think you are able to follow. Now coming into 24. Multiple alleles are 3. Then the number of genotypes produced will be same question, another form way I put it for question number 24. Same question, another form. Again, answer is 3. Formula here only I put it. So option is 3 for question number 24. 25. Regarding multiple allelism, choose the correct statement. Involvement of more than 2 alleles, right. These alleles are connected with the same character, right. In an individual, normally 2 alleles can occur, correct. In a population level, normally multiple alleles work. Population level only, multiple alleles work. So these all statements are correct. Option is 3. Now coming into 26. In connection with the controlling ABO blood groups, the plasma membrane of red blood cells has protein polymers sugar polymers Cell has sugar polymer, not protein, sugar polymer that protrude from its surface. And the kind of sugar is controlled by the gene. So option-wise, when we take it is 2, sugar and protrude, NCERT line. Now coming into 27. It is a biochemical oriented question, not directly in NCERT, but we can find it. In biochemical aspect, blood group belongs to. So each letter P, Q, R, S, we have to select P. When we take P, it should be IA because enzyme is N-acetyl galactosamine transferase, and only we can select it as IA. And recognize H antigen and adds to sugar part N-acetyl galactosamine, and we'll be able to get A blood group. Similarly, galactosyl transferase that belongs to B blood group, IB. H antigen, galactose, and forms B blood group. So for 27, answer is 2. Slightly in biochemical aspect, I put it. Now 28. If father is dash blood group, mother is dash blood group, suppose they have 2 sons and 2 daughters having AB, ABO blood groups, then what will be the blood groups of the father and mother? It should be like this way only possible, IA, IO, or reverse, IBIO. So we can get A blood group, IAIO. IO should be like this way only. IO, IBIO. So AB, then IAIB, then IO, IO. So we can select best option. It is heterozygous AN, heterozygous B in the sense IAIO, IBIO. So for 28, option is 2. This way problems will be there, simple problems. Reverse also possible. Now coming to 29. How many genes individuals can produce? How many gametes, not genes, how many gametes an individual can produce, human male? So no need to get confused. It is nothing to do with the number like a 2, 4, 8, 23, likewise, no. Nobody can predict it. How many gametes are the question, not the type of gamete like, say, capital A, capital B, capital C, capital D, capital E. Sorry, small e. How many types of gamete can be produced? Or small capital A, capital B, capital C, small c. This way take, this you leave. Here we can find 2, 2, 2. Suppose this manner. Only one type. Here is only one type. That is type of gametes. Here, not type of the gamete. How many gametes it is cannot predict. So option is 4. I think you got the question. What is a pair? So think about the question and do. 30. Identify the odd one. Odd one is Identify the odd one. Clue is Mendelism. What is the answer? It will be axial superposition because that is dominant. Remaining all are recessive. So option wise, it is 4. I think idea clear to all of you. If you like this episode, please subscribe or like it, share it, comment on it. We can meet later. All the best. Take care. Thank you.